Welcome back to Binary Bio, the channel where we bridge the gap between biology and computation. In this video, we're going to solve the counting point mutations problem from the Rosalind Bioinformatics problem set, also known as the HAMM problem. Before we jump into the coding, let's quickly understand what a mutation actually is. A mutation is simply a change that happens when DNA is copied. Because DNA carries instructions for how cells function, even a small change can have big effects. Most mutations don't matter much. Some can cause disease, and a very small number can actually be beneficial. Over long periods of time, these tiny changes are one of the driving forces behind evolution. The most common type of mutation is called a point mutation, where a single DNA base is replaced by another. When we compare DNA from two organisms, the number of positions where their sequences differ tells us the minimum number of mutations that could explain how they diverged from a common ancestor. In biology, we usually prefer the simplest explanation for evolutionary change. This idea is called parsimony. So, by counting differences between two DNA sequences, we get a simple estimate of how far apart they are evolutionarily. And that's exactly what today's Rosalind problem asks us to compute. The task is simple but fundamental. If I gave you two DNA sequences, how many mutations separate them? So, in bioinformatics, we often want to count how many positions differ between two sequences. That count is called the Hamming distance. The Hamming distance between two strings of equal length is the number of positions where the characters differ. Total difference is 4. So the Hamming distance is 4. That's exactly what Rosalind wants us to compute. We use a pointer I that starts at index 0. At each step, compare S of I and T of I. If they differ, increment counter. Move pointer forward. So we are manually walking through both strings in parallel. We are effectively tracking position in both sequences simultaneously using an index. We can also use zip st to pair characters from both strings. Then we loop through each pair. If the characters are different, we increase our counter. Finally, we return the total number of differences. This simple idea is used in comparing genetic sequences, measuring evolutionary distance, detecting mutations, sequence quality analysis. Even though this problem is simple, the concept is foundational in computational biology. That's how we solve the counting point mutations problem from Rosalind using Python. Now let's analyze the efficiency of our solution. Suppose the DNA strings have length n. We examine each position exactly once. We compare one character from the first string with one character from the second string. So if the string has n characters, we perform n comparisons. That means the time complexity is O of n. Now let's talk about space complexity. We only use one counter variable, a few loop variables. We are not creating any additional data structures proportional to n. So the space complexity is O of 1, constant space. If you enjoyed this video, make sure to hit that like button, subscribe to Binary Bio for more computational biology insights. In the next video, we'll look at another fundamental DNA transformation problem. See you in the next one.